There are two types of membrane receptors. The first is the G protein coupled receptors, and the second is the ligand-gated ion channels. The G protein coupled receptor lies on the plasma membrane. When a signaling molecule attaches, the G protein will come and bind to the GTP. If GTP is bound, then the G protein is inactive. When it is unbound, it activates and causes a cell response. The other one is the ligand-gated ion channel. The ligand-gated ion channel receptor acts as a gate for ions. When the signal molecule called the ligand binds to the receptor changes shape. With the gate open, ions such as salt or calcium can go through, thus causing a cell response. When the ligand leaves, the gate closes. The ligand-gated ion channels are very important to the nervous system. This is because ion movement through the channels may trigger an electrical movement.
What do you pronounce your title for the one that's the rates of paper being ripped?
Intracellular receptor proteins are found within the cytosol, or nucleus of target cells. These steroid messengers, testosterone in this case, are lipid-soluble and can cross the plasma membrane, a phospholipid bilayer. Only cells with testosterone receptors can respond to it. The hormone binds to the receptor protein and activates it, creating the hormone receptor complex. The active form of the receptor then enters the nucleus and activates genes needed for male sex characteristics. This causes DNA transcription and protein production.
The second step in cell signaling is transduction. This process usually involves multiple steps. It amplifies the signal sent by the extracellular signal molecule, as well as coordinates and regulates the cellular response. The main relay molecules are proteins. They activate each other in a set of chain reactions until the protein producing the response is activated. At each step along the line of proteins, the signal is transduced into a different form, which is usually shown in a shape change of the protein. Now we're going to talk about kinesis. This process begins to take place after the cell signal has been received by the receptor in the membrane and the relay molecule is activated. As I mentioned before, proteins are activated in a series of steps. However, how are they activated? This is where protein kinesis comes into play. It is the transfer of phosphates from ATP to protein, which can also be called phosphorylation. This changes the form of the protein from inactive to active. As you can see, this cycle continues until you reach the protein that signals the cellular response, and the series of protein kinases is called a protein kinase cascade. Eventually, you're going to run out of active proteins for protein kinases. However, the cell has a way to prevent this. Protein phosphatases removes the phosphates from the proteins, also called dephosphorylation. This turns off the signal transduction pathway, which allows the protein kinases to be reused for another signal that might be sent to the cell. There are also some kinases that are regulated by second messengers. The extracellular signal molecule that binds to the receptor is the first messenger. The second messengers are small, non-protein, water-soluble molecules or ions that spread throughout a cell by diffusion. Cyclic AMP and calcium ions are common second messengers. I'm going to mainly focus on cyclic AMP or cAMP, which is formed by adenylene cyclase, an enzyme in the plasma membrane, and it rapidly converts ATP to cAMP in response to extracellular signals. These then in turn activate protein kinase, and then it leads to a response. And this basically sums up how transduction works in a cell.
The response process of cell communication. Through the phosphorylation cascade, ATP interacts with adenylene cyclase, an enzyme in the plasma membrane that rapidly converts ATP to cAMP in response to a number of extracellular signals. cAMP then interacts with the inactive transcription factor called protein kinase. As protein kinase has two parts, which is catalytic, as you can see the green part, and regulatory, the red part of protein kinase, cAMP interacts with the regulatory part of protein kinase, which in turn then changes it from inactive to active. The active protein kinase in turn allows the synthesis of a protein by turning a gene in the DNA on or off, thus turning on DNA for transcription. rRNA, or ribosomal RNA, is transcribed from DNA and carries the genetic information from the DNA to be translated into ribosomes. Ribosomal RNA is the most abundant type of RNA, and along with associated proteins compose the ribosomes.